Okay, so in this video I want to talk a bit about the basics of proteins. And now you almost certainly recognise the word protein, and you'll probably know that they're one of the most important biological molecules. Really, they've got a huge variety in the human body, a huge number of functions as well. They act as they have structural functions. Perhaps best known for their enzyme activity, and a lot of this this functional activity, you can derive it from the huge variety of their structure. So I think what I'd like to do now is we'll go through a bit about their basic makeup, the basic structure, so that we can understand them a little bit better now. So start with the basics, as I always like to do. Proteins are made up from amino acids. You probably know this already, GCSE A-level biology, but let's refresh this now. These are the core building blocks of proteins, and so having a good understanding of them is really important to help us understand a huge amount of biochemistry and physiology, really. So why are they so important? Well, amino acids, that they have a similar structure. They all have a similar structure to each other. There's about 20 different amino acids that are common throughout all of biology, bacteria, human cells as well, and they're the building blocks of proteins. And whilst they have got some very similar characteristics, it's the differences between them that enable proteins to have such a wide variety of different functions. And I think that's easy to demonstrate if I highlight their structure to you now. So all amino acids, they have this basic carbon skeleton shape. You have a central carbon, an alpha carbon here, and it's got 4 different groups coming off it. So on the first hand, we've got the amino group. Unsurprisingly, they're called amino acids, and as you might guess, they've got an amino group and an acid group on them. And this is just a nitrogen and 2 hydrogens here. So that's your amino group. On the other hand, as we said, you've got an acid group, you've got a carboxylic acid group. So your carbon there, and the rest of the carboxylic acid group here. And that's the basic core structure of all amino acids. You've got a central carbon, you've got an amino group, and you've got a carboxylic acid group. You've also got hydrogen here, not very important. What's the next really important bit is that they have an R group here. R group just stands for the rest of the molecule, and as we can see here, this bit is the bit that stays pretty much the same throughout all the amino acids. There are some slight variations, but this is the core, the unchanging bit, if you'd like, of the amino acid group. Whereas up here, this is the bit that varies, and this is the bit which determines what different amino acids are. And there's a huge variety in these, and that basically determines, later on as we'll talk about, it's the R group that has an impact on much of the structure of the final protein molecule. So let's move on. We've got an idea of the basic structure of the amino acid there. But I think I should point out it doesn't actually exist in this form commonly in the human body. It exists as what's called a Zwitterion, in that, as we can see, both we've got an amino group here, we've got a carboxylic acid here group. They have acid-base interactions. The acid is unsurprisingly an acid; it's a proton donor, and the amino group is basic; it's a proton acceptor. And both these groups undergo that function at physiological pH. So we'll have the carbon here, and we've actually got you've got a COO minus group on that side. On this side, you've actually got a NH3 plus group there, in addition to the hydrogen and the R group. Again, this may be this is perhaps not seeming that relevant now, but this is important later on when we discuss the function a bit more. So yes, that is their overall structure up here, but in actual physiological pHs, they exist as as a Zwitterion with both sides with both of the functional groups ionised. Right, so we've talked a bit about amino acids now, and let's expand on this, and we'll talk a bit more about the proteins themselves. So what happens now? Well, we've got these amino acids, these are the basic building blocks, and we want to start to build a bigger structure out of these. And we mentioned the similarities between amino acids, and this is important because it allows them to polymerize, so it allows them to form these are the individual units that link together, and the most basic structure they form is what's called a primary structure, and that's just basically a long chain of amino acids attached to each other. I'll give you an example here. You've got one amino acid such as alanine. It will be attached to a cysteine molecule. It will be attached to another amino acid perhaps called serine, and this will keep going on. You have a long chain just in one row in a linear, in a line, and this is called the primary structure of proteins. Just a number of amino acids linked together in a chain. Also be called a polypeptide, just meaning from poly meaning many, and they're all linked together in a single chain. And I'll just go into detail now about the actual nature of these bonds really. So yeah, just to make it a little bit easier, I've made a copy of our molecule we drew before. There's one, and I'll play another one in here. Fantastic, there we go. And what we're going to do is we're going to talk a bit about what happens now. How do they work together to build this chain of a chain of amino acids, a polypeptide, and start to begin to get a basic proton structure. And I'll put them together close to each other like this for one very particular reason, and that's that these groups here are the groups that are going to react. And you've got one amino acid here, we've got one, perhaps a different one here. They've got this basic shape, but the R groups here might be very different from each other. But because they're all they've got a very similar core structure here, they've all got an amino group, all got a carboxylic acid group. So these opposing groups can come into contact with each other, and if you'll see there, all you've got is water. You've got H2O there, and what happens is that the water molecules, the hydrogen from the amino group and the OH from the carboxylic acid group come away, and what you get, I'll draw the structure out now just so you can see it. You get so you've got clearly got the different molecules. So you've got the alpha carbon of the first molecule, you've got your second carbon with the remaining oxygen from this group on this side, and from our second amino acid, I'll draw it in red. What we've got the nitrogen remaining with its one remaining hydrogen, and you've got the second carbon there with the other groups attached to it. And this here, this is what we've just drawn. That's called a peptide bond. And if you can picture here, that's bonding the two amino acids together. But you've got another amino group there. You've got another carboxylic acid group on this side, and you can form a long chain with multiple peptide bonds forming a polypeptide. So we can see here that you've got this going to have this carbon background I was talking about. Yes, a few nitrogens in there, but what we're going to have here is we're going to have lots of R groups sticking off this chain, and these R groups are going to interact with each other. These R groups can be quite big, and though we've got a single chain, as we've seen over here, we've got just a chain that's in one long row of different amino acids. You've got different R groups sticking off here, and we'll talk about now how these R groups can cause how interact in different ways to form to cause a shape of a more complex protein structure to form.